Hello everyone. InformationBoxTicket Lifestyles brings you today biochemistry topic on Gel Electrophoresis System, its apparatus, parts, types and examples. But before starting this video, don't forget to like this video, subscribe to my channel and press the bell icon button so you don't miss any of my upcoming videos. In the chemical process of electrophoresis, an electric charge in a solution flows in the direction of an opposing electrode. While studying blood proteins in the 1930s, Swedish biophysicist Arne Tiselius invented electrophoresis. Nobel Prize in Chemistry was awarded to Arne Tiselius in 1948 for his contributions to the electrophoretic method. One of the laboratory techniques for separating DNA, RNA, or protein molecules according to their electric charge or size is gel electrophoresis. We will learn in this topic its principle, its parts, the types of electrophoresis machines, operating procedures, applications, advantages of electrophoresis, limitation, precautions, and examples. Let's start with the principle of gel electrophoresis. The fact that the bulk of biomolecules exist as electrically charged particles with ionizable functional groups forms the basis of the electrophoresis principle. Depending on the pH, a solution containing biomolecules will have either positively or negatively charged ions. Charged molecules move in the opposite direction of the positive or negative pole when they are exposed to an electric field. Ionized biomolecules will migrate at various speeds in the presence of an electric field, depending on the mass and net charge of each particle in the solution. Positively charged particles move toward the cathode, whereas the negatively charged particles, such as nucleic acids, migrate towards the anode. Changes in the speed and direction cause each charged particles to move according to its unique attributes, allowing the separation of biomolecules components with related properties. Let's learn the parts of gel electrophoresis apparatus. With the help of this diagram, we will understand the apparatus. First comes the power supply. Constant current, voltage, or power are required for electrophoresis. Migration speed should be kept with a consistent power supply. The power source is connected to the gel box with the help of LED cables in the colors red, which is an anode cable which connects to the positively charged electrode, and a black, which is a cathode, which connects to the negatively charged electrode. These cables transport the electric current from the power supply to the gel box. Dissolved ions thermally stir as a result of increased resistance caused by an increase in the current. Equipment's water will evaporate more quickly. This will cause the ion concentration in the buffer to increase. The negatively charged DNA and RNA can go to the positively charged red wire at the front of the gel box by way of the black wire, which is linked to the back of the box. Then comes the buffer. The buffer establishes both the system's pH and the solute electrical charge. The following characteristics define the perfect buffer. Number 1. It should maintain the anylate's dissolving capacity. Number 2. Throughout the study, maintain a steady buffering capacity. Number 3. It shouldn't interfere with the ability to find the desired anylates. Number 4. It should obtain the proper distance between objects. There are two types of buffers, both an acetic and a basic buffer. Acetic buffers such as citrate, acetate, formate, and phosphate are used for a lower pH. To maintain high pH values, simple buffers like tris, borate, and tricine are used. The buffers have the same valency, ionic strength, and molality, are therefore made up of monovalent ions. When not in use, the created buffers need to be properly cooled since they might operate as a conducive habitat for the development of germs. The cold buffer can be used in the method since it improves sample resolution and decreases solvent evaporation. In the big volumes, the buffers can be reused up to 4 times. However, in small quantities, it can be discarded immediately away. The greater ionic strength of the buffer helps to provide a crisper resolution even if the considerable heat generated poses a significant danger of harming heat-liable compounds. Then comes the support media. Starch, polyacrylamide, agarose, and the cellulose acetate membrane in the shape of sheets, slabs, and columns are examples of supporting media. It is a colloid with a water content of greater than 90%. In order to separate molecules, it works as a molecular sieve. It is permeable, allowing small molecules to flow through but not bigger ones. There must be electrical neutrality. Nowadays, agarose gel is mainly used as a support medium for electrophoresis. Learn about the supporting media. Number 1 comes the starch gel. It is the first electrophoresis gel media. It makes it easier to separate proteins according to their molecular size and charge to mass ratio. Filling the starch granules solution in a buffer resulted in a colloidal suspension which, when allowed to cool, sets as a semi-solid gel as a result of the intervening of the branched chains of amylopectin. Use of petroleum jelly prevents swelling and shrinkage. It is possible to produce sharp zones and create resolving power. It is not currently employed since gel preparation with repeatability is difficult. Number 2 comes the cellulose acetate. Cationic acetate electrophoresis was initially created when Kohn demonstrated how to separate protein hemoglobin contained in red blood cells and to identify abnormal hemoglobin in blood serum. Cellulose in the filter papers is acylated to create cellulose acetate. Normal sites of acylation are at C3 and C6 of the glucose ring. The holes of the cellulose acetate are larger than those in polyacrylamide and agarose, two other popular electrophoretic matrices. Then comes the agarose. Agarose, a naturally occurring linear polymer made of galactose and 3,6-N-hydrogalactose chain, is present in agar recovered from red seaweeds. Agarose is stored as a dry powder similar to agar. By combining the agarose powder with the proper solution buffer, heating it and allowing it to cool to room temperature, agarose gel can be cast. The amount of agarose in a solution buffer regulates the gel's pore size. Agarose gels between 0.8% and 5% volume with weight are widely employed to differentiate between DNA and RNA molecules. Resolution is not as good as polyacrylamide gels. It creates stable gels, has a neutral charge, and has a low gelling temperature. As a result, it is regarded as the ideal substance for gel electrophoresis. It can either be liquid or solid. 4. Polyacrylamide. The co-polymerization of acrylamide monomers with the cross-linking agent NN-methylene bis-acrylamide, commonly known as bis-acrylamide, results in a clear, translucent gel. Acrylamide gel pores are regulated by a concentration of acrylamide which must be in proportion to its cross-linking agent. Usually takes a minimal quantity of acrylamide gel, which is 3% to 15%, to separate DNA and proteins. Proteins are separated under denatured circumstances according to their size using sodium didosyl sulfate SDS polyacrylamide gel electrophoresis SDS page, where a greater concentration of acrylamide gel 10% to 20% is generally utilized. Then comes the electrophoresis chamber. It is a plastic tank or container that is filled with a buffer to stop the flow of biomolecules. The migration process may easily be observed because of its translucent cover. It is connected to a power source. Number 6. Container for staining and destaining gel. In containers can be used for gel staining and destaining. Open foam boxes and closed boxes are also readily accessible. Typically, they are based on propylene. are fitted snugly and are transparent. They are stained and chemical resistant. Then comes the electrodes. Due to their capacity to attract charges with opposing charges, the two platinum electrodes aid in the separation of molecules. An anode binds positive ions, whereas the cathode binds negative ions. Lastly is the gel caster and comb. After being dissolved in the solvent, the gel is poured into a gel caster, which holds it and stores it inside the device. To position the wells for sample loading, a comb is used to arrange them. The types of gel electrophoresis. There are several types of gel electrophoresis, which are: number 1, paper gel electrophoresis; number 2, Agarose gel electrophoresis; number 3, Polyacrylamide gel electrophoresis; number 4, Pearls field gel electrophoresis; number 5, STS phage; number 6, 2D electrophoresis; 7, Immunoelectrophoresis, rocket electrophoresis. Lastly, difference gel electrophoresis. They may also be divided into two groups: native and denaturing. In native gel electrophoresis, RNA or proteins are maintained in their native form while moving across the gel. Contrast, denaturing gel electrophoresis reduces the RNA or protein to its linear form either before or during the electrophoresis process. By adding a reducing agent to the sample, gel, and/or buffer, this reduction is accomplished. This secondary structure is created as a result of separation of the bonds inside the RNA or protein molecules. Let's learn about the first, paper gel electrophoresis. Clinical context: it is employed to evaluate serum and other physiological fluids. Opacity and non-toxicity: it is easy to store. Absorption of proteins. Poorly conducting. Background staining: the OH group of cellulose bind to proteins and impede electrolytic movement, which results in bands, trilling, and poor resolution. So basically it employs filter paper strips soaked in buffer solution, usually diethyl barbituric acid and barbituric acid dissolved in alkaline veronal buffer, which has the pH of 8.6. A small volume of serum is placed on the paper and direct current is passed for several hours. Second comes the Agarose gel electrophoresis. The resolution of electrophoresis depends on the agarose concentration. It works well for isolating DNA fragments and sizes between 100 to 20 kilobase pairs. It also pertains to protein separation with the help of electrophoresis. Isoelectric focusing, which involves employing a low concentration of agarose gel, can be used to separate amphoteric molecules according to their isoelectric point. Third comes the polyacrylamide gel electrophoresis. Protein separation calls for a greater concentration than DNA separation and vice versa. It is utilized at a concentration of up to 30% with the pH of 4 to 9. A higher level of accuracy and dependability in porosity. Its uses in determining the molecular weight of DNA, sequencing DNA, examining DNA purity, analyzing recombinant DNA molecules, separating RNA molecules, and determining the molecular weight of RNA are all examples. Number 4. Pulse field gel electrophoresis. Parts and Contour first presented this approach in 1984. Adjusting the direction and intensity of the electric field between electrodes, DNA may be separated in agarose gel. This method is used to isolate higher molecular weight DNA with several megabases or even complete chromosomes. Because it generates exact findings that are easily reproducible, it is used in numerous disciplines. Used in research on a molecular biology of foodborne pathogens, monitoring the genetic stability of the organism, used in fermentation, mapping applications like chromosome rearrangement detection, RFLP, and DNA fingerprinting, and identifying related strains in the event of hospital outbreaks, among other things. 5. Sodium didosyl sulfate polyacrylamide gel electrophoresis, officially known as the UK Lamelli method after its British creator. Pore separating gel has smaller pores and a pH of 8, whereas the upper stacking gel has wider pores and a pH of 6. STS page, which uses sodium didosyl sulfate known as SDS, sodium lyryl sulfate, and polyacrylamide gel to separate proteins based on the polypeptide chain length, essentially eliminates the impact of the structure and charge. SDS, a detergent present in the sample buffer, along with a few reducing agents, cause harm to proteins' tertiary structure by rupturing their disulfide bonds. It is used to assess whether or not protein samples are pure or to compute the molecular weight of the protein. Isoelectric protein and isoelectric focusing. Proteins have no net charge. The pH level known as isoelectric point is reached. The high-resolution methods known as isoelectric focusing allows the separation of proteins by their isoelectric points within a continuous pH gradient. The outstanding resolving power allows separation of compounds with pI difference of about 0.1 pH units. It is employed to separate all amphoteric compounds, fractionate proteins, and identify isoenzymes. 6. 2D gel electrophoresis. It was developed as a combination of 2D gel, IEF, and SDS page processes and is used to evaluate complex protein mixtures. In the first phase, IEF separates the protein according to its charges, and in the second step, according to its mass. The IEF gel is placed horizontally inside the SDS page gel to conduct electrophoresis because SDS treatment renders the isolated protein on the gel negatively charged. Concentrated proteins on the isoelectric points are consequently split according to their molecular weights. Number 7. Immunoelectrophoresis, rocket electrophoresis. In immunoelectrophoresis procedure, the protein antigen is first separated using electrophoresis in semi-solid medium, and precipitin is then produced by immunodiffusion reaction with the antiserum. On the horizontal plate, suitable antibodies that are complementary to the test antigen to be assessed are dissolved in molten agar solution. Antigens are injected into the gel in the form of wells. At the alkaline pH, antigen gains a negative charge, travels in anode downward motion, combines with antibodies to form the antigen-antibody complex, and precipitates. The gel has been dyed with an appropriate dye like CBB, Comacine Brilliant Blue. The immunoprecipitates will then show as arcs like rockets. And lastly, difference gel electrophoresis, developed to deal with the quantitative aspect of studies on the differential expression and to overcome several problems with 2D phase such as analytical fluctuations. 3 distinct protein samples can be labeled with fluorescent dyes that are size and charge matched in order to observe each protein sample independently. For example, CY3, CY5, and CY2. Combining, loading, and putting the 3 samples through 2D electrophoresis. So let's learn the operating procedure of electrophoresis. Solution preparation. By dissolving it with hot water, a gel is created. The fluid is then put into a mold or caster after cooling to a more comfortable temperature. Gel casting. Once the gel has dried, well may be made in it using a comb. The electrophoretic chamber is then filled with the gel. Buffer can only occupy up to 1/3 of the chamber's overall capacity. Sample preparation. Dyes such as ethidium bromide or a fluorescent tag is introduced to the sample to give it color and density. To see how the sample moves across the gel, the DNA is extracted, pre-processed, and added to the solution containing some common blue dye. Sample loading. Sample is inserted into the wells using a sterile micropipette. Electrophoresis. The negative and positive LEDs which connect to the chamber and a power supply with a voltage is set respectively. When the power supply is turned on, an electric field and negatively charged particles are produced. Those molecules tend towards electrodes with the opposite charge to their own. DNA that is negatively charged migrate towards the anode, stopping electrophoresis, staining, and visualization. The dye is used to visually track the movement. Power source has been shut off. After the operation, the gel is dyed and seen with a gel imagizer. The logarithm of the molecular weight is used to determine the sizes of the sample fragments by comparing them to the standard. Application of electrophoresis. To investigate crime scenes and conduct paternity tests, DNA fingerprinting is used to isolate DNA fragments. Finding genetic changes and proteins connected to health and disease. For scientific applications, it is used in the detection and purification of nucleic acid and proteins. Finding infections in blood, other tissues, or sources like food is helpful. It makes it easier to identify and purify proteins or nucleic acid, where they are commonly studied in more detail using DNA sequencing or mass spectrometry. It is employed in evolutionary research and blotting techniques for the analysis of macromolecules. It makes polymerase chain reaction findings review more easier. Electrophoresis is advantageous for both the production and development of vaccines. Taxonomy DNA profiling is used to distinguish between species and evolutionary connections. Kindly show your support by subscribing to this channel. Advantages of electrophoresis. It is reasonably priced, establishes a relationship between outcomes that are comparable, very simple to carry out, can analyze DNA using any kind of proof, superior resolution, a wide variety of pore diameters are available, stable throughout a broad pH, temperature, and ionic strength range, transparent to light, chemical inactive, is both hydrophilicity and electrical neutrality. Limitation of electrophoresis. Number 1. Limited sample analysis. Techniques such as in situ hybridization can be used to examine the expression of genes at each individual location of the tissue sample. Every brain region in a sample can be examined using ISH, whereas only a few regions can be examined using electrophoresis techniques. Number 2. Measurements are not precise. Western blottings can effectively separate proteins with comparable molecular weight using gel electrophoresis. Using a technique known as 2D electrophoresis, it can also more accurately separate proteins. To determine the precise mass of proteins, mass spectroscopy must be performed after the protein has been purified. Number 3. A substantial starting sample required. Proteins cannot be amplified as DNA and RNA were able to do before electrophoresis. As a result, the efficacy of this approach is diminished by need for a larger tissue sample, and instead, flow cytometry and immunohistochemistry are usually employed to examine the protein expression in specific cells. For limited visualization facility, small hormones, neurotransmitters, and ions cannot be measured using electrophoresis. Do not completely respond to the electrophoresis preparation, often known as STS page, for two reasons, and even if they did, they won't be able to separate adequately since they are too small. Would hurriedly merge from the gel space. Number 5. Low throughput. Throughput in the sense that it takes a while to create data. Production of research data and the establishment of complex association by electrophoresis is subpar compared to PCR and flow cytometry, which are massively parallel and serial procedures. Precautions. Let's learn the precautions. Of non-conductive seats and flooring is encouraged, made of wood or plastics. When working near or adjacent to electrophoresis system, stay away from unintentional grounding points and conductors such as sink and other waste resources. Sample loading shouldn't be pushed too aggressively because doing so might damage the wells. While making the gel, put on gloves, a mask, and goggles. Consider taking the necessary measures before handling ethidium bromide since it is mutagenic and carcinogenic. Samples of electrophoresis system. DNA electrophoresis system GEP TH100TBT. Manufacturer is BioEvo Peak. Its features are: system has a built-in high current power supply that, by directly managing the current between the titanium anode and the stainless steel cathode, can immediately achieve high efficiency and fast transfer. The fastest and most effective protein transfer from gel to membrane is made possible by the transfer system's seamless integration of traditional transfer technologies. Focusing electrophoresis system BT10. Manufacturer is G Biosciences. Its features are: solves gel leak problems without tape. For small and big wells, there are two combs available, supplied with a power source that may be changed, easily operated and portable. DNA Electrophoresis System EPS 2014. Manufacturer is Innovia Lab. Features are: DNA and RNA electrophoresis. The EPS 2014 mini electrophoresis system has a small innovative design. Current can only flow to the electrodes while the lid is open due to a magnetic sensor. While the system is running, if the lid is taken off or open, the current is immediately turned off. And lastly, Isoelectric focusing electrophoresis system. Inofoni IEF, and the manufacturer is Hercuven. Features are: flexible device that can handle the majority of IEF requirements from low volume to high put applications. It functions with horizontal precast IEF and paste gels when the electrode frames is fastened to the cooling plate directly. So that's it for today. Don't forget to press the bell icon button so you don't miss my upcoming lectures. Thank you.